of Eastern Visayas State University, Burauen Campus. And now, we will be discussing about one of the most dangerous problems that is facing around the world, which is the coronavirus. And for information of everybody, that coronavirus is a time of what we will be discussing right now. And please allow me and please give me a little time to watch my video, to watch my presentation so that you can learn about what am I discussing right now about the virus.
If we will think critically, I know that we can form questions out of our curiosity. So maybe we can ask if where this virus came from, or when that virus was being introduced to us by our former scientists. So to give you a brief definition or a brief information about the virus, let us first start with its discovery. So it is stated here that in 1884, there is a French microbiologist, Charles Chamberlain, invented a feeder known today as the Chamberlain feeder or Chamberlain pasture feeder that was spores smaller than bacteria. Thus, he could pass solution containing bacteria through the feeder and completely remove them from the solution. While in early 1890s, there is a Russian biologist Dmitry Ivanovsky used this feeder to study what became known as the tobacco mosaic virus. His experiment showed that the extracts from the crushed leaves of infected tobacco plants remain infection filtration. While in 1899, the Dutch microbiologist Martinus Beijerinck observed that the agent multiplied only in dividing cells. And having failed to demonstrate its particulate nature, he called it as a contagium vivium fluidium. It is a soluble living germ. And in early 20th century, the English bacteriologist Frederick Dwork, who discovered viruses that infect bacteria. And another, in 1935, there is an American biochemist and virologist, Wendell Meredith Stanley, examined the tobacco mosaic virus and found it to be mostly made from protein. A short time later, this virus was separated into protein and RNA parts. And the breakthrough came in 1931 when the American pathologist Ernest William Goodpasture grew influenza and several other viruses in fertilized chicken eggs. And upon the discovery of the virus, there are three theories that was being introduced to us by our former scientists, and these theories were being used to prove that this virus is legit, that the discovery of the virus is 100% true. These theories are the regressive theory, the cellular origin theory, and the co-evolution theory. So let's start discussing what is a regressive theory. Regressive theory, it is the viruses may have once been small cells that parasited large cells, and over time, genes not required by their parasitism were hosts. The bacteria, rickets, and chlamydia are living cells that, like viruses, it can reproduce only inside host cell. And they lend credit to this theory because their independence on parasitism is likely to have caused the loss of genes that enable them to survive outside the host. And The cellular origin theory explains that some viruses may have evolved from bits DNA or RNA that escaped from genes of a larger organism. The escaped DNA could have came from plasmids. It is a pieces of DNA that can move between cells, while others may have evolved from bacteria. The last theory, the coevolution theory, explains that viruses may have evolved from complex of protein and DNA at the same time as cells first appeared on Earth and would have depended on cellular life for many million years. But like as humans, we commit mistakes, and these three theories that was being introduced to us leads to misconception and leads to disprove by most people. And these problems are the following. The regressive hypothesis does not explain why even the smallest of cellular parasites do not resemble for viruses in many ways. While the escape hypothesis does not explain the structures of virus particles, and the coevolution or virus first hypothesis contravenes the definition viruses in that they are dependent on host cell. I know that there are only few people who are totally knowledgeable, who are totally aware of what really a virus is. So I am here to discuss you about the entire definition of what totally a virus is, because I know that you are so confused by this moment that we are facing a pandemic right now. So I am here to give you the definition of a virus. Based on my research, a virus is an infective that typically consists of a nucleic acid molecule in a protein coat, and virus is too small to be seen by light microscopy and is able to multiply only within the living cells of a host, like the coronavirus. This virus is a very dangerous enemy because this is a very unseen enemy. We cannot see that virus, but that virus is transmittable to different hosts. Another is viruses are intracellular parasites partially the host cellular machinery for their own replication and offspring particle production. It is called intracellular parasite. Why? Because this virus depends only in producing, in reproducing, or in multiplying. They are only dependent on the host cell. Also, a virus is a collection of genetic materials and protein. And the place where viruses exist in nature act as a source of virus for hosts is what we call the reservoir. While the nucleocapsid is defined that the merge of the capsid and the nucleic acid together. So nucleocapsid is the sum of merging together the capsid and the nucleic acid. While the virion, it is a fully formed virus that is able to establish an infection in a host cell. And viroids explain that it is a highly wound circle of RNA without protein coat. Do you know what is the most interesting question about this topic? So I will ask you about, are viruses alive? Okay, so for my own understanding about the virus is not alive. virus will never and ever be alive. Why? Because virus does not metabolize. Virus does not respond to stimuli. And another, virus does not reproduce on its own. So it doesn't require the qualification of life. And even the virus is not alive, there are things that makes them alive. And what is that? Virus is a genetic material. It's either DNA or RNA, but not both. Viruses can evolve. Yes, it can evolve because it can be transmitted to another host. And viruses also classified similarly like as family or genus or species. Viruses have what we call the host cell. Does host cell die? And this is the biggest question. And do you know that there are two types of death? It is the immediate death and the delayed death. When we say immediate death, it is the lytic infection. Lytic infection is a virus enters cells immediately, then replicates, causes host cell to burst. And now it is a newly released viruses infect other cell. And another is the delayed death. A delayed death showed the lysogenic infection. Unlike the lytic infection, lysogenic infection is a genetic material that enters host cell, replicate with the host chromosome, and eventually switch to lytic infection, and they propage now in a bacteriophage DNA inserted into host cell DNA, and only few copies are made that will determine if infection became lytic. So when we say genome, the genome is the sum total of genetic information that carried by an organism. So it is the number of viral genes compared with a cell quite small. They only have the genes necessarily to invade host cell and redirect their activity. While the DNA viruses or the SSDWD, the single-strand DNA, and the dsDNA, double-strand DNA. While the RNA viruses, mostly single strands, positive sense RNA genomes that are ready for immediate translation into protein, and negative sense RNA genome has to be converted the proper form to be made into protein. And there are tools that are being used to study the structure of the virus. And what are they? What is that? The virus has a tool for electron microscopy. It is an excellent tool with some limitations. It is a highly resolution and image can bear distortion due to specimen processing. And another tool is the X-ray diffraction. It is good for naked virus and it has no envelope. And another is the cryo-electron microscopy. There are two types of structure symmetries. The first one is the helical and the second is the icosahedral. So let us first discuss what is helical structure symmetry. So helical is a rod-shaped capsomers and it is a symbol into helical nucleocapsid. Where the icosahedral, it is three-dimensional. It has 20-sided figure. It has 12 avenue-spaced corners, and although they all display the symmetry, they are wide variation. And now let us talk about the functions of the viral capsid. The functions of viral capsid, it protects nucleic acid and it helps introduce the viral DNA or RNA in a suitable host cell. Viral capsid also helps stimulating the immune system to produce antibodies that can protect the host cell against future infection. Unlike what I've said a while ago, there are many types of viruses, and I can give you example of this. The one that we are facing right now, the coronavirus. Another type of virus is the Ebola virus. Ebola virus is stated that it is a morphology under electron microscope. It is also a filamentous envelope RNA virus, and it is approximately 19 kb in length, and it is single-stranded, linear, segmented. Ebola virus is a negative sense RNA, and it is encoded in a 3 to 5 direction. It appears to have spikes due to glycoprotein on outside membrane. Another type of virus is the pox virus. A pox virus is a breed complex symmetry having 240 nm and 300 nm. It is also have an envelope that protects the antibody neutralization sites, and pox virus core contains enzymes for virus uncoating and DNA in inner membrane. Do you question out yourself that how virus can transmit from one host to another host? So like what I've said a while ago, viruses can multiply or can be transmitted from one host to another host. But the question is how? So at this moment, I will give you the processes, the viral processes of how the virus can be multiplied to the different hosts. And these processes are the following. First is the attachment or the adsorption, followed by penetration, and another is the uncoating and synthesis, and the final processes is the assembly and release. So now let us first talk about what happened during the attachment or the adsorption. In attachment and adsorption, viruses encounters susceptible host cell, and it absorbs specifically to receptor sits on the cell membrane. Because of the intact fit required, viruses have a limited host range. And in a penetration process, flexible cell membrane of the host is penetrated by the whole virus or its nucleic acid. It is endocytosis because the entire virus engulfed by the cell and enclosed in a vacuole or vesicle. The viral envelope can also directly fuse with the host cell membrane. While the process in uncoating, the enzymes in vacuole dissolve the envelope and capsid. The virus is now uncoated. And in synthesis process, free viral nucleic acid exerts control over the host synthetic and metabolic machinery. DNA viruses enter host cells' nucleus where they are replicated and assembled. DNA enters the nucleus and is transcribed into RNA. The RNA becomes a message for synthesizing viral proteins, and it undergoes the process of translation. The new DNA is synthesized using most nucleotides, and RNA viruses replicated and assembled into the cytoplasm. And the next process is the assembly or the release. In assembly, mature virus particles are constructed from the growing pod of parts. And in release, non-envelope and complex viruses are released when the cell lysis ruptures. Envelope viruses are liberated by budding or exocytosis, and anywhere from 3,000 to 100,000 variants may be released depending on the virus. The entire length of the cycle anywhere from 8 to 36 hours. So that is the process on how the virus can be multiplied to another host. So there are many ways to cultivate a virus, and we will talk first the primary purposes of viral cultivation. First is to isolate and identify viruses in clinical specimens. Next is to prepare viruses from vaccines. And the last is to do detailed research on viral structures, multiplication cycles, genetics, and the effect on the host cells. In using live animal inoculation, especially bred strains of white mice, rats, hamsters, pigs, and rabbits. Occasionally, invertebrates or non-human primates are used. Animal is exposed to the virus by injection. In using tissue culture technique, most viruses are propagated in some sort of cell culture. The cultures must be developed and maintained. Animal cell cultures are grown in sterile chambers with special media. Cultured cells grow in form of a monolayer. It is a primary or continuous. There are classification. Viruses are classified on the basis of habitat, the host, which is trivial system. Besides, the viruses are classified on the following criteria. It is classified by the structure, the chemical composition, similarities in genetic makeup, International Committee on the Taxonomy of Virus, which includes 3 order or 3 variants or 263 genera virus. So there are 3 types of classification: the Daltimore classification, the classical system classification, and the genetic classification. And the last topic that we will be discussing is the benefits of viruses. So, in genetic engineering, harmless viruses are used as genetic vectors which carry good genes into cells. Viral envelopes stimulate the immune system to produce antibodies that can protect the host cell against future infections. Viral genome contains enzymes for specific operations within the host cell. Antiviral drugs block virus replication by targeting one of the steps in the viral life cycle. Interferon shows potential for treating and preventing viral infections. Some recently developed drugs do combat some viruses, mostly by interfering with viral nucleic acid synthesis. AZT interferes with reverse transcriptase of HIV. Cyclovir inhibits herpes virus DNA synthesis. So, even if the virus is a very dangerous enemy, it is a process of fighting off infections. The first is the antiviral drugs that interfere with enzymes' genetic information. However, body rarely ever is cured from virus because drugs kill host cell as well. And another fighting off of infection is due to vaccination. Vaccination teach body how to fight infection when they see it again. And the first vaccine was developed in late 70s by Edward Jenner is to fight smallpox.